Hello, everybody, and welcome to the second part of the MPAN session. The first speaker of this second part is Professor Miguel Sena Esteves from the University of Massachusetts Medical School, and his talk is entitled Gene Therapy for MPAN. So, Professor Sena Esteves, please, the stage is yours.
All right, well, thank you for the introduction. I think I modified the title a little bit since I sent the title, but the intent is exactly the same. So first of all, thank you for inviting me to give this presentation. I'm going to...
Dr. Sena, I'm sorry to jump in. Just asking Dr. Tiranti to turn her camera off, please.
Oh, okay.
Don't think she can hear me.
So I think you should just turn her camera off in that situation.
There you go. Okay, thank you. All right, so the goal for today is to give you just a little bit of a background in adeno-associated virus gene therapy, and to discuss a little bit of a pathway towards clinical trial based gene therapy clinical trial for MPAN. So adeno-associated virus is really a tiny, tiny virus, probably one of the smallest ones in existence. It's a 20 nanometer capsid made out of protein, so it's not an enveloped virus. It's a single-stranded genome, DNA genome with 4.7 kb. It has 2 genes, wrap and cap, and it codes 8 proteins. So in the context of a virus, it really is tremendously simple. And it's called adeno-associated virus because it actually was discovered as a contaminant of adenovirus. And importantly, it's also one that for its replication, it depends on other viruses, and hence being part of a family called dependoviruses. Down here, you see an electron microscopy, not quite as well as adenovirus. It's a little smaller particle, and here is the first 3D structure that was determined about AAV2. Over many years, the trick has been to really find a way in which to replace all the genetic information, and that's really how all AAV vectors are made, of supplementing in trans the wrap and cap genes during production. And essentially all there is inside the virus is a transgene cassette that has the elements necessary to express the gene of interest. And here is just some electron microscopy on the left showing empty and full capsids that usually result during production. So there are several aspects that make AAV vectors quite attractive for in vivo gene transfer, one of them being the very high efficiency of gene transfer, albeit requiring relatively high dose or very high dose in some cases. It does not integrate into the host cell genome. For the most part, it stays as an episomal element. And there is really evidence that gene transfer and gene expression is a lifelong lasting event, which of course has advantages to it, but also can pose serious concern about safety in case something goes wrong. So one has always to plan these trials in the context of having a situation that you cannot go back and pull it back out of the individual, like a small molecule that you stop taking it and there is a wash period, and after that you're presumably fine. So you have to enter into a clinical trial relatively carefully, or actually quite carefully, taking that into consideration. In terms of CNS delivery routes, and while Maciej talked about the peripheral symptoms and the potential for peripheral disease associated with MPAN or mutations of the C19orf12, certainly the leading cause of the phenotype or the most severe phenotypes in these patients, to my limited understanding, is a neurological disease. So we'll focus today perhaps talking about this, and certainly some delivery modalities can both treat the CNS and the periphery. So where the field started with gene therapy was essentially having the ability to use intraparenchymal infusion, where viruses were directly infused to particular targets. Techniques were developed like convection-enhanced delivery to enhance the distribution and create more uniform distribution fields for transaction of cells. And eventually we moved on to CSF delivery and systemic delivery due to the discovery of new capsids that have quite interesting properties. So the notion of an intraparenchymal delivery certainly is designed to target specific structures. Convection-enhanced delivery that has been pioneered by Chris Benkowitz at UCSF, as well as other groups at NIH, have enhanced our ability to cover entire structures in the human brain. And it turns out that some products, such enzymes in lysosomal storage disease, can actually move through axonal transport as well as growth factors. But in addition to that, using very high doses, in fact, vectors themselves can also move throughout the brain through axonal transport. And so that creates two approaches to achieve broader distribution of gene transfer in the brain in addition to the target structure. There are ongoing several clinical trials, some of them for Parkinson's disease, targeting the caudate and the putamen, and there's also for lysosomal storage diseases. In terms of systemic delivery, in 2009, there was a watershed finding that AAV9 can actually cross the blood-brain barrier. So after systemic administration, so the blood-brain barrier prevents free passage of most molecules or macromolecules into the brain and regulates tightly what it allows to cross the blood-brain barrier. But it turns out, for reasons that we actually don't fully understand, AAV9 can cross the blood-brain barrier and transduce cells in the central nervous system. And this was a paper from Kevin Faust and Brian Casper in 2009 that really led to a revolution in our field. And in addition to demonstrating gene transfer in mice, which of course is a starting point, they also went on to demonstrate that, in fact, if they treated monkeys, they could actually deliver genes throughout the brain quite consistently. You can see here on the left side an immunostaining showing expression of a marker throughout the entire brain, and in the control, of course, no staining. Now, an important finding to consider in the context of a gene transfer and gene therapy for a disease like MPAN caused by C9orf12, that is a mitochondrial protein, is that the transduction in the postnatal stage occurs in the brain mostly in glia. So this is one of the limitations of the systemic gene transfer, except in the spinal cord, where it actually transduces spinal cord motor neurons quite effectively. Nonetheless, and this, of course, this finding that in fact it transduces motor neurons very effectively was used to develop a therapy for spinal muscular atrophy, and in 2019, again, a landmark approval from the FDA for a product. And I have nothing to do with the Avexis or Novartis, so this is an endorsement of their product. This is really stating that this product is now approved for use in the United States, and it's in different phase. I think it's approaching approval in European Union as well. The delivery is through CSF. Most of it has been done through intrathecal delivery. Again, it can, several types of AAV capsids actually work. You can transduce quite effectively spinal cord motor neurons, and it's shown here, again, immunohistochemical staining for a marker, and showing here on the bottom left, transduced motor neurons. In many instances, one of the limitations of this approach is that you can't really deliver the gene to neurons in the cerebrum at very high efficiency. And I draw your attention particularly to the deep brain structures, and what you can appreciate, and I'm going to try to make the mouse, actually, I'm not sure if you're seeing the mouse. I hope you are. Oh, let me see the spotlight. There we go, the spotlight. Oh, it's not allowing me. Okay. So anyway, you can see here in the figure A, the fact that you see transduction, so cells that are expressing the marker gene in the cortex. You can see higher magnifications here on the bottom. But one thing that you can appreciate in panel A is that the deep brain structures really are left almost untouched. So this is something that you have to consider, one has to consider in using an AAV9 for a CSF delivery approach. So in the context of MPAN, there is a number of considerations, and these are from my somewhat limited understanding of the biochemistry and certainly the neurological and disease presentation. But there's a couple of facts that I think are important to consider. One of them being that C19orf12 is a mitochondrial protein, and I learned this morning from Angela that in fact, it's a protein localized on the outside of the mitochondria and linking the mitochondria to the ER. One of the issues with mitochondrial proteins, perhaps not necessarily one that is on the surface of the mitochondria, is that often overloading mitochondria with a protein can actually be quite toxic. And while in short-term assays that may not be the case, in cell culture, in the context of neurons, that could be a major issue. The other aspect that Susan talked about earlier is her finding that in fact there are dominant forms of the mutation. So many are recessive, quite a few that are dominant as well. So that should be taken into account. And then the question of targeting the entire CNS, the entire body. We know that of course the protein is missing everywhere, but then one has to consider where the state of gene therapy is right now, the state of the art, and whether one should consider targeting basal ganglia, given that that's where, at least based on MRI, a lot of iron storage takes place. So in terms of thinking about how a program, a gene therapy program could look like, so the first two things to take into account is it's a mitochondrial protein. Overloading could be toxic. So one, in my experience, should look into a range of potencies that one would have to test, given that at the moment this is mostly an empirical process. And these could be done relatively easily in wild-type mice, and really trying to, at the molecular level, looking at single-cell RNA-seq, so looking at the single-cell level and trying to compare the amount of mRNA that is made from the endogenous gene to that is made from the vector, and really trying to match them as closely as we possibly can. Our field in general has taken this approach of using extremely strong promoters, in a way a little bit of a Lego type of approach in it, or plug and play, if you will. But I think we're also finding more recently that that may actually be a problem for certain proteins, and I would say that for a mitochondrial protein, one has to approach it relatively carefully. And one can test various delivery modalities, such as intrastriatal delivery modality, can test systemic delivery, et cetera, et cetera. There's a number of modalities that one can test. And the question of, and this is Susan's paper that she actually mentioned during her talk, the genetic mechanism of recessive versus dominant. It's clear that for the dominant diseases, gene supplementation alone is not adequate to treat these patients, given that these patients already have a normal copy of the gene, but certainly seems that the mutant protein has a dominant function. So one can have two approaches. Certainly one can focus on the recessive mutations, and I'm not sure why this is not showing up here. Okay, well, one can focus on the recessive mutations alone, but certainly will leave some patients out of the equation, or one can actually develop a dual function AAV vector where it simultaneously silences the endogenous gene and expresses a new one. I have a little graphic here showing essentially the concept of using microRNAs where they can be expressed. The microRNAs are produced through the endogenous mechanisms. They target the mRNAs, and certainly what will happen is that all mRNAs can be degraded, and for some reason the presentation is not working properly, also degrading the gene that you encode into the AAV vector. However, what you can do instead is actually design an mRNA expressed from the AAV vector that is resistant to a microRNA that is now become specific to the endogenous gene alone. And what you'll have, in fact, is that you will continue to make normal protein, but you now have degraded the mRNAs from both endogenous alleles that you are trying to replace by the one that you're bringing in with the AAV vector. So the advantage of this is developing allele-specific silencing. It's possible, actually quite difficult as a matter of fact. It is certainly that a lot of people are working on. But the limitation there is that then you would have to develop an allele-specific approach for every single patient or different mutation that is out there. So instead of doing that, you can actually silence the expression of both alleles and re-express a normal protein based on what you are encoding from the AAV vector. So the The path forward, essentially developing a dual function AAV. This is something that a colleague of mine has pioneered at UMass, and it's relatively straightforward, consisting of identifying microRNAs that are highly efficient or hyperfunctional to silencing the endogenous mRNA and eventually protein. One would have to then engineer a cDNA that encodes that is resistant to the microRNA by simply wobbling the coding region, and there are particular regions in the microRNAs that are more sensitive to mismatches. And essentially, you then conduct studies, that safety studies in normal mice, and move on, accompanied at the same time for studies in patient cell lines to restore stress response, given that at the moment we don't have a mouse model where to actually test the function and therapeutic efficacy of mice. This is not necessarily a major limitation to development of a clinical program. In terms of, again, developing a clinical program, one can take it into two different ways. Certainly one approach is that targeting one of the areas where we know there is neuropathology in the basal ganglia, whether neuropathology in the basal ganglia is a major driver or the unique driver. The likelihood of that is, I would say, relatively low, given that it is a protein that is expressed everywhere in the body. The advantages of a targeted approach is that there is considerable experience in Parkinson's disease clinical trials of AAV gene therapy. The delivery is well tolerated. There is no side effects from it. The techniques that Chris Pankowitz has developed really lead or allow us to cover the targets at very high efficiency. Another aspect that is very attractive is the fact that one does not need extraordinarily high doses for each patient, and therefore mid-scale productions can actually be used to treat lots and lots of patients, so decreasing the cost of a clinical trial and eventually a therapy. The cons of this approach is that, in principle, it's a local effect, maybe spread out to some cortical areas through axonal transport of the vectors. But nonetheless, this is not an approach that would cover the entire CNS. And that raises the question of, in fact, whether one would be able to alter the disease progression or stop it altogether by targeting the basal ganglia alone, and perhaps even the substantia nigra. Of course, the other approach is either systemic or CSF delivery, where the goal is to treat or deliver the gene to the entire CNS. One of the things that this will enable is to potentially impact peripheral phenotypes, since systemic delivery through the vasculature would naturally transduce quite a few tissues, including the liver, muscle, heart, etc. CSF delivery does that to a certain extent as well, because a lot of virus actually escapes to the periphery. Now, as I've told you earlier, the cons of this approach certainly is the low or relatively low gene transfer in the brain. We're talking about a relatively small percentage of neurons that are transduced. Also, the fact that deep brain structures that clearly are part of the pathology or the neuropathology of MPAN are not transduced very effectively. It's spotty transduction at best. There is a potential for toxicity. That's something that we've learned relatively recently when we need to hit very, very high doses. We've learned that the painful way, and more so for the families of the kids that died recently when treated with doses at 314 per kilogram, which you can imagine at 314 per kilogram. Now the requirements for treatment become enormous. And so the scales of production to even run a clinical trial are very significant, that raise the costs quite high. In terms of how could a program look like, this is maybe split it in two phases. One before engaging the FDA and EMA, where it consists a little bit of the steps that I already described before, and essentially combining that with alongside of clinical trial design by close interaction with the clinical experts on these diseases, obviously lots of them neurologists. And then after an interaction guidance from the one or both regulatory agencies, the experiments would be planned to conduct the IND-enabling studies, and then engage in figuring out how to manufacture GMP-grade AAV vector for the clinical trial, as well as for the IND-enabling safety studies, although you don't necessarily need GMP material for a phase 1-2 trial. You just need to make it in the comparable method. Engage immediately in the IND preparation to try and reduce the time between completion of the studies and submission to the regulatory agencies for approval of phase 1/2 trial. So I think with that, I am done. I think I'm more or less on time, and I'll be happy to answer questions that you may have. Thank you.
Thank you very much, Dr. Sena. So let's start with questions. So can you say something about how you avoid an immune response if the patient had already been infected by natural AAVs?
Well, so that does present a bit of a challenge. It represents a challenge, first of all, for well, depends on the route that you're going to implement. So if you do try to have a systemic delivery, patients that have already been exposed to AAV, they usually have neutralizing antibodies that may prevent you from actually treating them. If patients have been exposed prior to the treatment to AAV, certainly it is possible to keep them under an immune suppressive regimen. In fact, that's something that several clinical trials have done. And there is evidence, in fact, that that may be necessary for an extended period of time to prevent such immune responses as the question. So that was us. So really, we at the moment really don't have a way to bypass the immune system. And in fact, when people talk about AAVs that the immune system doesn't see or won't react to, I think it's people are fooling themselves, because after all, it's always an antigenic protein. So we try to deal with it in the best way that we can, which is essentially immunomodulation after the fact of the gene transfer. And I think that's all we have at the moment for human.
We have a second question. Would you expect that one would need to combine gene therapy with other drugs that in addition would remove the iron that was already built up?
Hard to say. I mean, I think without having animal models that reproduce the disease, including the iron storage, I mean, it's really hard to know for sure, right? I mean, I guess we can draw some parallels to lysosomal storage diseases, that it's not accumulation of iron, but it's accumulation of other metabolites of gangliosides, lipids, glycosaminoglycans. And the reality of it is that once you restore the enzyme function in a cell, you can actually clear it. Whether metabolism of iron is involved, or C19 or 12 is involved in direct metabolism of iron, I actually don't know. So I don't know if it's possible to restore it. And, the combination with other drugs, I think, at least in the initial stages, that's almost guaranteed that would be an advantage to have a small molecule combined with gene transfer. I don't think at this moment we're at the stage where one could say, well, yeah, gene therapy will take care of everything the first time we try. So I think regardless of whether he clears it or not, I think that need will be there nonetheless.
Okay, and the final question. Could the microRNA potentially have off-targets effect?
Absolutely, sure. That is certainly a possibility. It's something that one can, so the genome, obviously a mouse genome is not the human genome. Even a monkey genome is not the human genome. This is part of a process that tests safety ultimately in humans, and that's part of the reason why you should be fairly conservative about how you plan a clinical trial, because the last thing that you want is really take a situation that is already pretty severe and make it worse by essentially killing a patient. So that's something that you have to approach carefully. One would naturally try to look at the transcriptome of animals that have been treated and enrolled in a safety study. There is evidence that microRNAs, at least we've published a paper with colleagues here at UMass showing that if you use siRNAs that target very effectively some genes, for instance, Huntington's disease or HTT, And in fact, there's really no evidence of alterations, at least in monkey, no evidence of alterations of the transcriptome. So based on that, it is possible that they won't, but this will require screening of microRNAs. But that's a great question. Thank you.